We are going to talk about the sensory systems of the termite and other insects. You'll notice a lot that there isn't always a lot of information on termites, so we often also compare them to cockroaches. So the nervous system of the termite consists of a cerebral ganglion, the subesophageal ganglion, the thoracic ganglia, and the abdominal ganglia. So the central cerebral ganglion controls the behavior of the termite and processing. The SOG controls the salivary ducts, the muscles around the head, thorax junction, and the mandibles. Different types of termites actually have different central nervous system structures. So soldiers are going to have a much larger SOG and posterior nerves than worker termites do. The cerebral ganglion connects to the ganglia that are spread throughout the periphery of the body. A ganglion is made of multiple nerve fibers, which are a large central nerve cord called a neurophile in the center. The Sensillium are the sense organs and are often hair-like structures attached to the exterior of the termite that perceive specific stimuli such as vibration or touch. The Sensillium connect to the endings of associative neurons, which connect to the neurophile, which connects to the central ganglion for processing. Notably, the insect nervous system is much less centralized than that of vertebrates, so we don't have a clear distinction between a central nervous system and a peripheral nervous system.
The strength of termite vision depends on the role they have in their colony. Reproductive termites can see. This includes the king and queen as well as swarmer termites. Reproductive termites are tasked with reproducing and even leaving the colony to start a new one. Worker and soldier termites are not able to see. These termites can call on their other senses to do their roles for the colony. Cockroaches have compound eyes, which is a visual organ consisting of thousands of ommatidia, which are tiny, independent photoreception units that consist of cornea, lens, and photoreceptor cells, which distinguish brightness and color.
While termites do not have a nose, they do use their antennae for olfaction purposes. On the surface of each antenna are multiple pore tubules that are able to collect pheromones and other chemical signal molecules. Inside the antenna surface, there's a single micrometer-thin water-based fluid layer known as sensolar lymph. This is what chemical signals molecules must cross before reaching the olfactory nerve and chemoreceptors. Because the sensolar lymph layer is water-based, the odorant signal molecules have a difficulty crossing it because they are insoluble in water. Because of this, odorant binding proteins, or OBPs, bind to the signal molecules and coat them in a water-soluble layer. This allows them to effectively cross the aqueous barrier and reach the olfactory receptors. There's actually very little information regarding the tactile sense of termites. Nonetheless, termites do participate in a form of vibrational long-distance communication, which suggests some form of tactile receptors as a means of communication. Termite soldiers perform a head drum, which is literally just smacking their head against a substrate while under duress to create a vibrational alarm signal. This can be received by nestmates throughout the colony, as they have a threshold amplitude of 0.012 meters per second squared. The vibrations are thought to be received using microsensory hairs that range in length from 8.084 millimeters to 0.179 millimeters, and termite soldiers overall contain a larger sense lobe when compared to other termite roles, picking up signals more efficiently, which indicates that increased sensitivity to vibration stimuli is more important for colonial defense. While microsensory hairs are able to be isolated, there was no information to be found regarding the mechanism of mechanoreception in termites specifically.
So the information regarding thermoreception in termites is also very limited, so we researched cockroaches. Cockroaches use their antenna as a thermosensory system, and they only have one receptor type called the cold receptor, which is used to provide both cooling and heating information. They are excited by cooling and inhibited by warming. And the cold receptor axons converge into 3 fused glomeruli, forming a structure called DC1 at the posterior margin of the antennal lobe. From here, there are cold projection neurons, which exhibit excitatory responses as a result of cooling, and warm-cold neurons at the cold glomeruli. The warm receptors exhibit excitatory responses in response to warming, and the warm-cold receptors exhibit excitatory responses as a result of warming or cooling. From the antennal lobe, the axons of the neurons project to the olfactory and thermosensory areas of the lateral protocerebrum, which is part of the brain where the signal can be processed.
Just like many other insects, termites do not have physical ears. However, they are able to detect vibrations through sensors in the base of their antennae and the tibia of their legs. When these vibrations are detected due to disturbance or occurrences such as recruiting nestmates, termites exhibit a behavior known as head pounding. Head pounding is the act of drumming the heads against the substrate. and creating a pulse vibration. These drumming signals are trains of pulses of substrate vibrations with a pulse repetition rate of about 20 Hz. The signals transmitted travels only a short distance, but enough that the whole termite community is aware.
Spatial location isn't well studied in termites, but we can look at cockroaches. Cockroaches use sensors in their antennae to detect the polarization of light. Their antennae are also sort of used as walking sticks to make contact with external surfaces to guide the termite. This information from the light polarization is sent to the central ganglion, which processes this information to determine the location of the insect in terms of the amount of polarized versus unpolarized light that is present, as well as based on the contact with the external surfaces. Interestingly, cockroaches are not able to process an internal representation of their spatial surroundings, but instead rely on the understanding of the lightness versus the darkness to navigate where they are.